Welcome back. We just finished the mitochondrial round. Now it's time to talk about CRISPR, the newest, latest, hottest technology in science. Luckily today, one of our contestants, Giovanni, here is an expert in CRISPR. Now let's go talk to him. Hello, sir. What do you know about CRISPR?
Okay, so what is CRISPR? CRISPR is a gene editing device. This is where the genome is fundamentally altered by cutting DNA strands and replacing them with other DNA. CRISPR edits genes by precisely cutting DNA and then allowing natural DNA repair process to take over. So basically, when the Cas9, one of the CRISPR system's enzymes, locates the target DNA, it bonds to it and cuts it, thereby turning off the targeted gene. And basically replacing the gene code can change the physical and biological features of an organism.
Thank you, Giovanni. That was some very inspiring work you shared with us today. Now, let's get on to Jeopardy! In the lead, we have Selena with 6,000 points and a four-way tie for second, with Nikita, Giovanni, Ashley, and Zara all tied at 5,000 points. Remember, each contestant has 30 seconds for their response. Selena, let's get to the Jeopardy! Round. Okay, question. What does CRISPR do?
What is hijacking genes to make new genes and embed them?
Correct. 200 points.
Using the Cas gene, which makes protein that shifts CRISPR around when needed to modify genes. Sequence of gene can edit the entire DNA, inactivating specific genes that could be harmful. Also, CRISPR could edit the gene by adding a host DNA, and would fill itself in.
That's correct. That's 200 points for you, Selena. The board is yours.
Thank you, Marcus. I'll take overview of CRISPR for 300.
Who invented CRISPR?
Who is Jennifer Doudna and Emmanuelle Charpentier? They are considered to be the mothers of CRISPR. Jennifer Doudna was one of the first scientists to propose CRISPR as a microbial mechanism for genome editing. She changed the world for programmed gene editing and found potential to fix near infinite number of problems in the genome. Co-founder Emmanuelle Charpentier is a French professor and microbiologist. She paved the path to CRISPR using past experience with microbiology and found its inner secrets. CRISPR was discovered in 2012, and both scientists recently got a Nobel Prize in 2020.
That's correct. Ashley, you have the board. Ashley, that's correct. You have the board.
Marcus, can I have history of CRISPR for 400?
Jennifer of the University of California at Berkeley and Emmanuelle Carpenter of the Institute in Germany were awarded the Nobel Prize 2 years ago for being the first to describe CRISPR and how to theoretically edit DNA. In a patent battle, the U.S. Patent and Trademark Office agreed with Broad, changing CRISPR's commercial future as well. But it was the researchers from the Broad Institute of MIT and Harvard who were the first to actually do it, publishing their own study a year later, proving that they could successfully modify mice and human cells. Also, due to the licensing of the CRISPR technology, it makes it super difficult for certain biotech companies to create gene editing therapeutics based off of CRISPR.
Action. Correct.
Marcus, could I have structure of CRISPR for 400?
Sure thing, Giovanni. Your question is, what are the two main parts of CRISPR?
What are the Cas9 protein and guide RNA? So the Cas9 protein contains lytic functions that cut and or paste new DNA segments into the target cell's DNA, whereas the guide RNA is what tells the protein complex that it's arrived at the target destination. The guide RNA is a sequence of nucleotides that are complementary to the nucleotide sequence on the target gene. So once the guide RNA successfully binds to the DNA, the Cas9 protein snips off either end of the target sequence, and if a new sequence is to be added to fill in the empty space, RNA polymerase is what synthesizes the respective sequence. And then the ligase goes in and it seals the new fragment onto the template DNA.
Correct, Nikita. You have the board.
Thank you, Marcus. Can I have structure of CRISPR for 200?
How is CRISPR made? Well, genome editing is a specific type of technology on how scientists change DNA. CRISPR-Cas9 is made of two key components. The gRNA, which is guide RNA, and the Cas9 protein. The Cas9 protein, Scientists identify a DNA strand that needs to be altered, and they they make a specific guide RNA to that specific DNA to alter the DNA sequence with the Cas9 protein cutting and unwinding. This is how scientists use to edit or delete specific parts of DNA.
Correct. 200 points to you. You have the board now.
Can I have cost of CRISPR for 500 points?
What is $2 million? So the monetary cost of CRISPR varies depending on the type of procedure one wants to undergo. But currently the treatment for a disease such as sickle cell anemia, which is caused by the malformation of red blood cells as a result of a genetic mutation in the HBB hemoglobin gene, that treatment costs about $2 million per patient. But less invasive procedures such as genotyping, on the other hand, cost upwards of about just $1,200. That being said, despite the hefty price tag, the CRISPR gene editing procedures, they are far more accurate than some of the other leading genetic treatment technology.
That was correct, Nikita. Now it's time to cut to a commercial break. See you soon.
Have you ever just thought of genetically modifying your child? Well, I got some good news for you. With the power of CRISPR, you can. For starting around $16,000, you can make the child of your dreams. You want to choose gender? You got that. Choose physical traits? Of course, that's part of the package. The future with CRISPR, it's in your children.
Action.
Side effects of CRISPR. The gRNA leads to the wrong DNA strand, which causes Cas9 to edit the wrong portion of DNA. This side effect used to be more common, but the improvements have gotten more precision in the gRNA function. Double-strand breaks that are caused by CRISPR can lead to genomic rearrangement due to the individual chromosomes being shattered. Improvements of technology. Researchers have developed a new method which makes CRISPR gene editing more precise than conventional methods. The method selects molecules best suited for helping CRISPR-Cas19 protein with high precision editing all correct location in our DNA. The gRNAs come in different sizes and shapes, and not all of them are equally effective. As a result, the challenge is to identify which one works efficiently, which is what the technique accomplishes, which was created using new information collected by researchers. This also predicts that gRNAs functions the best. The approach, which has been successfully developed, combines our own data with publicly accessible data, which includes understanding of molecular interactions between gRNA, DNA, and the CRISPR-Cas9 protein.
Welcome back from commercial break. Here's a quick word from our champion today, Selena.
CRISPR has unlimited potential with people who want to do research. The possibilities of being able to fix genes, a person that has irregular genes that regulate their function. If CRISPR can somehow change the gene that inhibits the proper function of proteins, that can save many lives.
Well, that wraps up another great episode of Jeopardy. Thank you and have a good night. See you soon.
CRISPR makes corn crispier.
Sup, y'all? I'm an agricultural scientist. So how does CRISPR make our corn crispier? You see, CRISPR is gene editing technology. We can use this to edit the gene in the corn, and the corn will be bigger, taste better, and will produce more. Keep supporting agricultural CRISPR use.